Hello, everybody. Welcome to Genomify. Today, we're going to be looking at part three of the cut and run data analysis series, and we're going to be focused on performing peak calling on your cut and run sequencing data. If you've processed and aligned your cut and run data, but you're struggling to figure out how to call peaks, you are in the right place. Um, Today, we're going to be focusing on a software called SEACR to perform our peak calling today. So, if you're just joining me now for the first time, there are a couple steps you need to have already performed for this video to really make sense. So, previously, you need to have already aligned your raw sequencing reads and performed a normalization factor calculation from these BAM files. You also need to have filtered your BAM files and performed normalization and bedgraph conversion. If this doesn't make any sense, please go watch part one and part two. It'll get you up to speed. However, if you've already done all this, we're going to jump right into the conceptual overview of what we are going to do today. So, if you are unfamiliar with peak calling, you may be asking, what exactly is peak calling? So, when you run a cut and run or ChIP-seq experiment, you get back millions of sequencing reads scattered all across the genome, but you don't want to know where these reads are mapped. You want to know where your protein of interest is actually binding. And peak calling is a computational process that takes those raw reads and identifies specific genomic regions with statistically significant enrichment. It's basically asking, where do I have way more signal than I'd expect by chance? These enriched regions, which are called peaks, represent the actual binding sites or regions of chromatin accessibility if you're looking at ATAC-seq data. And so on the bottom here, I'm just showing a track of our bigwig files, and here are the peaks that SEACR called on these particular bigwig files. I like to think of peak calling as something a human could really do. It's just doing it across the entire genome. So when you're looking at your sequencing data, anything that looks like a peak should be called as a peak from your peak calling software. That's a good gut check I always do with my peak calling analysis. I always double check it by eye. And so the real challenge is distinguishing true biological signal from background noise. In this case, that'll be an IgG background control. And that's where these different peak calling algorithms come in. And so today we're going to be using a software called SEACR, which is specifically designed for cut and run data analysis. So see, here is how SEACR actually works. First, it takes your cut and run sample. Let's say our target is SMARCB1 here. And then you have your IgG control, which represents your background noise. SEACR scans both data sets and identifies what it calls contiguous blocks. These are essentially just regions where reads are back-to-back showing signal. And you can see here, if you point to the red brackets, there are 6 regions of contiguous signal. And then if you look at our IgG sample, you can see that there are 7 regions of contiguous signal. Now here's the key. Some of these targets overlap with the IgG background, which suggests that these might just be noise where the MNase is cutting by pure chance, while others are unique to the actual target that we're looking at. So SEACR asks the question, at what threshold do we best separate real signal from the background? And it does this by testing different total signal thresholds and plotting what fraction of these blocks are filtered at each different threshold. So the red curve shows the target blocks and the yellow curve shows the IgG blocks. And as we increase the threshold, as we go from left to right, both curves drop because fewer blocks have a signal that exceeds this signal threshold. But notice how the target curve drops much slower and maintains a higher fraction of these blocks as we go to the right. And this is because our target signal is actually stronger. And so SEACR, what it does is it finds the point where the ratio between these curves is maximized. As you can see in this arrow, this is the part where the distance between these two lines is greatest. And this is where you're keeping most of the target signal, but you're also excluding most of the IgG signal. So now when we apply this threshold, here's what happens. So some peaks meet the threshold and are kept. These are our high confidence peaks. However, there are other target regions that are filtered out by this threshold. And these can be either too weak, meaning that the threshold cuts them off, or they're overlapping with the IgG. So we know that those are likely just background. And really that is the beauty of Seeker is that it's not using these arbitrary cutoffs or using Poisson distributions. We're just assuming that your data is giving you the best threshold to give you the best signal to noise separation. So Seeker gives you two different filtering options. You can either do stringent or relaxed mode. So stringent, if you look at this light blue um, line, is denoting the maximum distance between the red and the yellow line. That is also matching to the black arrow over here. And so this is where you're going to get the absolute best discrimination between your target and your background signal. These are your high confidence peaks. And it also has this other option called relax mode, which uses a slightly lower threshold set halfway between the knee or the region where the kind of direction of the line changes and the optimal peak. So it's essentially just shifting this arrow a little bit to the left, halfway between this black line and this curve here. And so this is going to capture more peaks, including weaker ones that might still be biologically relevant, but you trade off a little bit of specificity for sensitivity. And so for my SMARCB1 variant study, I used the stringent mode because I really wanted the high confidence binding sites for functional validation. But if you're doing more exploratory work or you happen to have weaker signal from your cut and run experiments, the relaxed mode might actually be better. All right, so you may be asking, Garrett, why do we not just use Max 2? Everybody uses Max 2. And that is a fair point. Max 2 has been the go-to peak color for over a decade now. But here's the thing. Max 2 and Seeker have completely different philosophies. And so I'll break it down one at a time. So Seeker takes a more empirical approach. It looks at your actual data and asks, what's the cutoff that gives me the best discrimination between the target and your control? There's no assumptions, there's no statistical models, there's just optimization based on what you actually measured. On the other hand, you have Max 2, and this program uses a lot of statistics. It assumes that your reads are following a Poisson distribution and runs a hypothesis test for all particular regions. And what it's asking is this region significantly enriched above the background IgG? And if yes, it'll output a p-value under 0.05 or 0.01, or in this case, it'd be a q-value in FDR, and then boom, that's called a peak. So what is the problem with Max2 in cut-and-run data? So cut-and-run data gives you incredibly clean data, actually, with very little background noise, and that's really one of the biggest advantages of using cut-and-run over a technique like ChIP-seq. And that's amazing, right? Well, actually, though, because it has such little background, it really breaks the Max 2 statistical model. So when your background is basically zero, even two or three random reads in your target in a spot will look statistically significant compared to nothing. So Max 2 goes crazy and calls all these peaks and regions that really have no enrichment at all. There's just a couple reads that happen to be aligning there. And so Seeker doesn't really have this problem because it's requiring your target to be strongly enriched over the IgG, not just statistically different from a near background of zero. So that's why we use Seeker for cut and run instead of Max 2. Okay, and just taking a quick programming detour, this is something we're going to be utilizing in our Seeker command to iterate through multiple sample types and their associated IgG control. And what this is called is an associative array. So most of you have probably used regular arrays before. This is just an ordered list where it says like 1, 2, 3, separated by brackets. Associative arrays are a little bit different. They store pairs of information. You can have a key and you have a value. And these keys can have multiple values associated with them. As you can see, this file names underscore wild type underscore rep1 has 5 different lines within it. And so this phone book has a kind of nested text file associated with this key, and it also will have an attached value. And this is really helpful when you're trying to link a control to multiple input files. And so I like to use a classic analogy like a phone book. When you look up someone's name, that's your key, and you can get back their phone number, which is the value. And in this case, multiple people can have the same phone number. So you can have like multiple different samples attached to the same IgG control. And so in my peak calling script, I'm going to be using this associative array to create this lookup table. And my keys are this text files, which list all the different sample names associated with each genotype. And my values are the corresponding IgG control files. So basically, this just helps the command be maintainable and you can update it very easily if you bring in more samples. And this just prevents us from having to type the same plain old secret command like 40 different times, which is just super annoying. And it just makes the code generally cleaner. And so you'll see what I mean later when we look at the code. Okay, so just taking a quick broad overview of what we're going to be doing today. First, we're just going to simply call peaks. Step two, we're going to filter peaks for a minimum signal threshold of 5, and you'll understand and see why I'm doing that later. And lastly, we're going to just be finding the overlaps between the two replicates that we sequenced. And so we just want to keep the peaks that we are confident and have been seen in two different replicates. And so before we jump into the code, just gonna walk through what you actually need to have installed on your instance to perform this analysis. So first, you need to have R version 4.4.3 on your instance path. You also have to have SEACR source code, which you can download from their associated GitHub page. And these need to be downloaded and put directly onto your instance, ideally the directory that you're going to be running this command in. You also need to have two programs, GNU Parallel, and another one called bedtools. I put that here. And this is going to help you find the union of all subunits to find consensus, SNF complex binding sites. And you'll see more what I'm talking about later when we go through that. All right, so now that we've covered the conceptual side of things, we're going to jump into the coding part now. All right, so before we get into the actual analysis, we first need to go obtain the source code for SEACR and actually pull that onto our AWS instance. So what I'm showing here is the GitHub page associated with the SEACR publication, and all you really need to do here is download this file right here, as well as this file right here. And so it's pretty straightforward, but all you're going to need to do is just click this. Click download. We'll come back here and download this one. So now we have our two SEACR commands downloaded onto our computer. And then once we've downloaded these two files, we will upload them to our AWS S3 bucket and then move those two files onto our instance, preferably the instance we're doing the analysis in. And so I've shown how to do this in previous videos, so I'm going to kind of assume you already know how to move files around on the AWS environment. So now that you have these on your instance, we can actually begin with the analysis steps. Okay, so picking up where we left off on at the end of part 2 of this series, but here we are working in this process BAMS PE directory, and we have already generated our normalized bedgraph files. And just quick refresher, just to show you what these files actually look like. It's essentially breaking the genome into these bins and has an associated normalized signal value for each of these bins. So that's going to ultimately be the input for our SEACR peak calling analysis. And so there are a couple things we need to do first before we can actually run our peak calling command. So first, we need to ensure that we have our in our path already set up in our instance. And the way you do this is just typing in which R. And so most AWS instances already have this preloaded into the path. So just a good gut check just to see if it's there. If it is, SEACR can do what it needs to do. Another thing that we will need to do is create these file name text files. And I'll kind of explain what I am talking about here. So for a cut-and-run experiment, in a given cell line, you're going to be probing multiple targets. So in this case, I was probing H3K27, H3K4, SMARCA4, SMARCB1, and SMARCE1. And this was all in one cell line expressing a specific genotype, I315I, which is a silent mutation. And so for each of these marks, you have an associated IgG background control just for this cell line. And so our sequencing data actually ends up having an IgG control for each of our conditions tested. So we have essentially, in this case, 6 IgG controls. And we need a way to link these respective controls to their experimental target conditions. And so we're going to utilize these these text files essentially. And I just created one for each of the replicates across all 6 conditions that we tested within our cut-and-run experiment. This really is just going to depend on what you did in your experiment. If you only used, I don't know, 2 genotypes and only had 2 replicates per genotype, you're only going to have 4 of these text files instead of the 12 I have here, because I did 6 conditions and 2 replicates for each. So I'm going to have 12 text files to represent the different controls and their experimental conditions. And so what we're going to do once we have these generated, and I'll just jump right in and show you the actual peak calling command here. Okay, so this is the entire peak calling command, and I'm going to walk you through what each of these steps is actually doing. Again, this is really just a text file I created in this instance. You can copy and paste this exact same text as long as your file names, text files have your file prefixes correctly, and you rename this IgG normalized bed graph to what your IgG normalized bed graph is called. This should all work just fine. But conceptually, I'm going to walk through what each of these commands is actually doing. Okay, so this top little bit with the declare a controls, this is creating an associative array. So like I had explained in the PowerPoint, you have in an associated array, you have keys that have an associated value. And so what we're going to do here is we're going to provide the key here in this first part of this command separated by brackets. This is just telling Bash, this is our key text file. And then we are connecting this key with a specific value. In this case, it's an IgG background control. And so what this array essentially is telling Bash is that this control is associated with all of the lines present in this file. So what this computer is going to do is read in each of these lines sequentially. And it's going to say, for this prefix, this is attached to this IgG control. And then we'll move to the next line and say that's also connected to this same IgG control, so on and so on. And so we're just creating this large array of samples and which IgG file they are connected to. It's very straightforward. Honestly, I know it seems a little bit confusing, but trust me, this is just how this works, and it makes the code much more simple. Okay, so then what we're going to do is use a for loop to iterate across every single line that is present in our associative array. And so to do this, we're just going to use a for loop, and we're going to assign each of the keys, in this case, all the values present on this left side, to a variable called file. It's going to loop through each of the lines present in this text file, so on and so on and so on. It'll just kind of go through these sequentially. And then what we will also pull out from this associative array is the control. And so the syntax here, it's not super terribly important, but I'll explain it anyway. So in this case, you can notice that there's an exclamation point right here. This is essentially telling Bash to pull out the key from this associative array, aka the left side, and assign it to this file variable. For this control, you see how the exclamation point is missing. That is telling Bash to pull out the associated value for this key. And so that's essentially just going to copy this file name to a variable called control. And so this is just saying we're processing this particular target with this particular control. And so you can actually visually see what is running when this command has been run. And then we're going to be utilizing parallel to process each of these lines sequentially. So essentially what's going to happen here is it's going to pull all 5 targets from this file, this text file, and compare them to this IgG control. So it's basically going to do 5 processes simultaneously. And that is what parallel essentially lets us do, is to run all 5 of these processes in parallel, which just makes the analysis much faster. And so really what's just kind of going on here is this parallel, and then this is just telling it, we're providing it the inputs file path to actually run within this command. So we're just providing it the, what is this? The file prefix, and it is going to be found in our processed bams.pe directory, the one up here. And so this is just providing the prefix and then the path in which that file is present. Here I'm telling it that you can run 8 jobs at once. You really only need 5, but I just bumped it up to 8 just to be careful. And then this just ensures that the output from different jobs don't mix together. And so this just ensures that our output is actually kept in tidy order and each parallel process is assigned to a single output. And so the way that we're actually going to run SEACR is by calling the source code, this SEACR underscore 1.3.sh. So we'll just type in bash, but we're just telling bash to run this particular text file. And then what it's going to do is we're providing the target normalized bedgraph file. And so what this A from parallel does is it inputs this essentially list of letters, this whole path plus the prefix, and puts it right here between these 2 squiggly brackets. And then this will tell bash the rest of the file name. So we're essentially just providing the path and the file name for our target normalized bedgraph file. And then we're providing our control through this control variable. And that is going to be exactly what we put here in this value column. And then this is going to be a little specific to this particular data set, but SEACR offers an in-house normalization strategy. And if you don't provide non here, it's going to try to perform its own normalization. Since we've already done our spike-in normalization previously, we're just telling SEACR, don't worry about the normalization. We've already taken care of that. So it's important to include this non if you've done E. coli normalization already. And so then, like I had talked about in the previous slide deck, in this case, we are using the stringent filtering threshold, but you can also change this to the relaxed if that is what your data seems to want. So I like to do both stringent and relaxed and just compare them. It's no harm in doing both. And then here we're just providing the output file. And so again, I've created this directory called SEACR output in the same directory that we're working in right now. And then it's just going to add this list of letters. This is essentially just a variable of letters, and it's going to paste it right here, just as it had done previously, right here. And it's just going to add this suffix called SEACR peaks at the end, just so we know what each file actually is. So it's ultimately going to be the peak file is assigned in the SEACR output with the suffix SEACR peaks, and each will have its own respective prefix. And so then this will just echo that once it's run through every single of these keys, it will say, great, we're already done with this file. So you should get however many lines are in your key text files. In this case, it would be 12 times 5, which is, so I'll have 60 lines telling me which one it is processed. And when it's done, it'll stop running, and then we can actually go and look at the output. And so here I will, oh, it bummed out and timed out on me, but we'll look at the SEACR outputs in just a second. Just a quick disclaimer, I realized I didn't show you how to actually run this command, so it's really straightforward. All you do to run this command and make sure it doesn't time out if it takes a while is type this nohup period forward slash peak calling to, and then add this ampersand. And when you hit enter, that will run the command in the background, and you will actually be able to see what files are being processed each time as it's running through the for loop. And so I just wanted to put that in there. I realized I forgot to put that in the previous video. So yeah, that's how you run it. Okay, so now that we've actually outputted our peak files, I just wanted to walk through what our actual output from SEACR means. So I'll just open one of these really quickly and just explain each of the columns so you know what you're actually looking at when you finish this. So when you are looking at your SEACR output, you have 1, 2, 3, 4, 5, 6 different columns. So the first column is going to be the chromosome or the contig that this particular peak was called in. This is going to be the start, this is going to be the end, and this is going to be the total signal across the entire peak. What this is actually showing you is the most intense signal within this peak. So this is looking at one tiny region, this is looking at the signal across the entire peak. And this is going to give you the specific genomic region that is encapsulated within this summit, as SEACR calls. So ultimately, if you want to know how many actual peaks are in a given sample, all you need to do is just hit more, copy that, and just do pipe word count L. So in this case, this has a ton of peaks. This is H3K27, so that makes sense. If you want to look at something more like this, you can see it's going to have substantially less peaks because H3K4 trimethyl has inherently less enrichment in the genome than H3K27. And so we'll kind of go through how to iteratively process each of these samples, look how many peaks are in each, and that will help us guide what additional filtering steps we will need to do for these peak files. All right, so I moved my camera up a bit so you can see what I'm actually typing here. But at this point, we have all of our SEACR peaks called for all of our conditions. And you might be thinking, great, we're done. Let's go do some annotations, make some heat maps, and see what everything looks like. But hold on. We need to talk about why we can't just use these raw peak files as is. Let me show you the problem. So if I count the number of peaks in each file, and so the way I'll do this is by using a for loop where I assign each bed file to a variable, and then we're going to loop through these and count the number of files or lines in each. And we're going to echo, and then we'll do this word count command, and we're only going to keep the actual number that comes there, and then put the actual file name outside in a tab after. And then we're going to save this to this file here. And then we're going to look at the actual target that was giving us a problem, which was SmartB1. So we'll just filter this for anything that has SmartB1 in it. All the other ones actually look pretty okay, so I'm just going to show you what was going on with the SmartB1 protein here. Okay, so we see that something weird is going on here. If we look across all the different genotypes, we see that the wild type, or in this case the silent, has anywhere from 3 to 20,000, which isn't a huge jump. This is not totally unexpected. And again, we see a lower range for our missense mutation. But something interesting is going on here with our nonsense mutation. Why are there 100,000 or 200,000 peaks being called in a nonsense mutant? And I'll remind you, this I315X is a nonsense mutant, meaning a truncated form of SMARCB1. You shouldn't really have any binding sites because there isn't a full-length SMARCB1 there. And so the real question is, are these I315X peaks actually real? And is this actually meaningful biology, or is the algorithm just being too permissive here? Okay, and so now to answer this, we need to look at the distribution of signal across all the different peaks. And see that these are all just super weak peaks. And I have a suspicion that it is teaser. They are. And so what I'm going to do is I'm going to create a distribution and bin it by bins of 5. So I'm reading only the bed files with SmartB1 in them. And I'm going to use the same thing where I have a for loop and assign them to a variable f. And then I'm going to echo them like I had done before and save them to this text file we create called SmartB1 histogram summary.txt. And now we're going to use an awk command to perform this division and assign it to an integer in terms of bins. So I'm just going to set up my awk command here. I'm going to print the 5th column and divide it by 5 and take the nearest integer. And so int, so column 5, which is where the signal value is, divide it by 5. And then we're just going to multiply that by 5. And then we'll close that off and print the actual file name next to it. Then we're just going to sort these so it's lowest to highest. And then make sure we don't have any overlaps. And then we will echo it again. I didn't do, I gotta finish this off. The for loop wasn't closed. So we'll let that run. And so if we look at this distribution, we'll see something interesting. So, okay, so let's look at each of these different samples here. We can see that for I through 15I, the average signal is around 20. If we look at the second replicate, we get ranges all the way from 25 to 55. And again, if we look at our missense mutation, we get anywhere from a couple peaks in the 45 to 60 range. So here is the problem. While all of our other all of our other samples have signal values greatly exceeding 10, our weird sample, this I through 15 stop, had almost all of its peaks below a signal value of 5, which is shown here. The vast majority of the peaks being called have a very, very low signal. So, look, what's happening here is that SEACR is using this adaptive thresholding. It looks at your data and says what's enriched relative to the background in this sample. It's comparing your IgG to your target. It doesn't use a universal cutoff. So for this nonsense I315X, because the entire sample has a weak target enrichment, SEACR sets a very low threshold. Anything marginally above the background gets called as a peak. That's why we have greater than 100,000 peaks, but realistically, they're actually all just noise. This isn't really biologically meaningful. They have a very low signal. So for all the other ones, the algorithm is seeing a clear signal and sets a higher threshold. If you remember the graph that I showed you in the PowerPoint, there's a greater separation between the two farther down that threshold. But for our nonsense, that maximum is a very, very, very low threshold, meaning the IgG and this SmartB1 target in the nonsense were very, very close actually. So only the strong biologically meaningful regions get called in the other ones, but in our nonsense mutation, tons of background noise is being called. So what's the solution to this? We need to perform hard filtering. We can't let the algorithm be the only judge. We need to impose a minimum signal requirement based on what we know is reasonable for what our cut and run data should be showing. So if you think about it biologically, I through 15 star or X is a nonsense mutation. There's a premature stop. We expect loss of function. That's exactly what we're actually seeing. There's low signal, there's no specific binding, there's just background noise everywhere. On the other hand, I315I up here is synonymous. This should be a functional SMARCB1 protein. We see focused, strong peaks at specific genomic loci. That's what functional chromatin binding looks like. So we need to apply a hard filter to our bed files. And when we do this, we're actually doing two things. For all the other samples, we're pretty much just cleaning things up. We're removing marginal peaks that snuck through. And I recommend doing this for any cut-and-run data set, really. But for our tricky samples, we're honestly asking, are there any real peaks here, or is this actually just complete loss of function? All right, so let's do this filtering. I'm going to set a threshold of a max signal greater than 5 to keep only peaks that exceeded that signal threshold of 5. That's pretty conservative, but I'd say it's reasonable for cut and run. So to do this, we'll do the same for loop we have been doing. We'll read in all of our bed files. We'll do a similar awk command that we've been doing. We'll take the 5th column, and we'll only keep anything that is greater than 5. And then we'll keep the file name. And then we will create a new bed file called filtered. We'll close off that for loop. I didn't close this. And so once this has filtered everything for 5, we're now going to look at the number of actual peaks in each file that exceed 5. So we'll do a similar thing for I in this case, we'll now only look at the filtered bed files that we just created. We'll do the same command that we did earlier. Well, we're just counting the words for the lines, and then having another tabbed column, and just saving it to this filtered counts.txt file. This is a quote, not an apostrophe. And so now we'll look at this, and we'll grep any ones with SMARCB1. All right, and so now that we see we have filtered a lot of the peaks that were being called peaks without this hard filtering threshold. And we can see that the other ones, we didn't really filter any peaks in here. And so I will say that we are still retaining some peaks. I didn't want to be too hard on my filtering and bump it up to 10. But in our next step, we're actually going to be combining peaks that are observed in both replicates. And that will get rid of most of these background peaks here as well. So really, probably we'll only have 4 peaks that are held because those will only be found in both peaks. And just to convince you how this is a terrible thing, you can see that the rest of these are likely just barely above a signal of 5 when every other sample 5 isn't even on the radar. So when we actually end up doing the heat maps or really any other analysis, these are going to come up as relatively not significant. So this is really just an artifact to make sure you don't have way too many peaks in your cut and run data. And if I wanted to get rid of these, I could bump up the filter to something like 10, but then I might actually start filtering off some biologically important peaks up here. So there's a trade-off, and you have to kind of pick your battles and see which ones you want to filter. But ultimately, because we're only looking at peaks that are observed in 2 replicates, it should be fine as we go forward. And so we'll get into that in the next step. All right, so at this point, we have filtered peak files for both biological replicates, rep1 and rep2. And a critical question we need to answer is, which peaks are actually reproducible? So when you're looking at your final cut and run data set, You want to really only focus on peaks that are overlapping between replicates, as these are most likely to be true biological signal. If replicate 1 calls a peak at a specific site, a good peak should also have the same peak called in replicate 2. That's how you know a peak is real biology and not just noise or technical artifact. So we need to compare each rep1 file with its matching rep2 file and find the overlapping peaks. That's what this next step does. So here is the script. I'm not going to type this all out because it would take forever. But let me walk through what it's actually doing. So first, we're just going to make this replicate overlaps directory just to make sure there's actually a place to put all the output of this. Then what we're going to do is we're going to loop through all of the rep1 files, pretty much every replicate 1 peak that we have already filtered. And then for each of these rep1 peak filters, we're going to assign it to this variable called r1 underscore file. And then for next, what we're going to do is extract the prefix attached to this r1 file variable. So if the particular file is called, for instance, i315x, mark E1, rep1, yada, 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 we really only want the prefix. We don't really care about all of this. So the way that we do this and extract the prefix is just use this double percent sign, and that's just saying cut off anything that looks like this, and whatever's left, put that into the prefix. And so that's going to help us read it into the rep2 part. And so then we're going to assign these prefixes in addition to the rep2 suffix, which we'll find up here. And we're just going to basically stitch the prefix with this suffix and add it to this rep2 file. Okay, so now that we've created all our variables, we have our rep1 file and our rep2 file. This is really just helping the for loop do what it needs to do. We first need to just check and make sure this file actually exists, so there's not like a weird analysis going on. Essentially, it's saying if it does exist, do this. If it doesn't, then output there's an error. There shouldn't be an error, but I like to put these things in there just in case something wrong is going on. And so here, what is actually happening, I guess before I do that, we actually have to create this output variable as well. So we have the rep1 file, rep2 file, and the out variable. And in this case, we're just going to tell where we want to put this output. So we're going to put it in this replicate overlaps directory, use the same prefix, and then we're going to give it the suffix that we created called rep1 versus rep2 overlap. And then so bedtools here is really doing the heavy lifting. And so pretty much what we're saying is that we're going to take the rep1 file as A, and then take the rep2 file as B. And then we're going to find any regions where they overlap. And then we really just want to save both the signal values for the first rep and the signal values for the second rep. So we're going to keep everything. So we're going to have like a weird file structure that I'll show you in just a second. And we just want to read it out to that output variable that we've described here. And so essentially, this is just a for loop that's going to run through each of the rep1, rep2 pairs. And the way that you run this is the same that you run it for everything, but you'll just do no hub, period, / find overlaps, ampersand, hit enter. I'm not going to do that now because it takes a little bit of time, but ultimately everything would be outputted in this replicate overlaps directory. And that is what these look like. And so now that we have the regions where the peaks overlapped, but the output of the command isn't exactly what we want. So if we look at what is actually in here, let me zoom out here so you get a better idea. You can see that we have essentially two different columns. We have all the information for rep1 and all the information for rep2. We don't really need this. All we really care about are what are the genomic regions that are present in both peaks. You can't really average the two signals together. That's not something that you typically would do. So we're just going to extract specific columns from these rep1 versus rep2 bed files, and that will ultimately give us our final consensus set of peaks. Okay, so I'm going to quickly cover conceptually how we're actually going to be merging peaks between the two replicates. So as I showed you before, the actual output of the previous command looks something like this, where you have the peak signal data both for replicate 1 and for replicate 2, and it shows you the start and end of those peaks in each replicate. And so if you were to plot this one line, for example here, for one peak across two replicates, you can see that the peak for rep 1 actually has greater min and a greater max, or less min and a greater max than the peak called in replicate 2. And so the method that I used in this particular analysis was just taking the minimum across both replicates and the maximum across both replicates and kind of combining those signals together to get the greatest absolute coverage for a given peak. This is a relatively permissive form of combining peaks between replicates. If you wanted to be more conservative in your peak calling, you could take only peaks that are identified in both replicates, so you would end up with a merged peak that looks something more like replicate 2. But in this case, since both peaks are really being called in both replicates, I thought it would be okay to just expand it to the minimum and maximum observed in both replicates. So you can do what you want in your own analysis, but I'm just showing you how I actually did this in my own analysis. So now we'll jump into the code and show you how I actually did this. Okay, so now that I've described the actual output we're working with, which can be seen similarly shown right here above, I'm going to show you the actual code we use to identify the overlaps between two replicates. And so I'm using a for loop here to iterate over all the wild type or silent conditions. This can be expanded to include other genotypes as well, but for this particular study, I was mostly just interested in where are the consensus peaks for either the wild type or the silent. And then we can use this kind of master list to then compare signal values at our mutant uh, genotypes. So I'll kind of walk you through what this command is actually doing, which is I called it make reproducible peaks.sh. And so here pretty much I'm just creating an output directory called master list. I always put this at the beginning of these commands just to make sure there's actually somewhere to put my output. I'm creating these two variables, one genotypes and the other marks, and I'm pretty much just applying the genotypes I want to apply as well as the marks that I want to analyze here. And I'm just using these as placeholders when I'm creating my input variable within my nested for loop here. So I'm pretty much just using a nested for loop here to pull in all the different genotypes, in this case only two. And then for each of these genotypes, do another for loop where I create this variable called mark. And so ultimately what's going to happen here is it's going to take the genotype variable and the mark variable, and I will then construct the name of the file. For instance, here in this case, it would be I315I genotype, and then I'd put H3K27 trimethyl for the mark, and then rep1 versus rep2 overlap. This is essentially just making it so our input files actually look like our actual file names. So in this case, this is what I am trying to create here just within a nested for loop situation. And then similarly, I'm just going to create another variable. The only thing that's really going to be different here is I'm going to input it into this new directory I created up here, and I'm going to add this different suffix here, just showing that these are the reproducible peaks across replicates. And I always just put in this kind of echo just to show what sample is actually being processed while this is running. And so then the way that we're going to actually calculate the min and the max, as well as pull out the chromosome value, is using this awk command. And so awk is a pretty nifty tool. If you're not familiar, definitely try to learn it. It's really helpful in Bash. But essentially, all I'm going to be doing is I'm creating this awk command, and I'm going to include a ton of different steps. And this right here is essentially telling awk what is the input. You always put the input after in an awk command like this. And that file name will be held in this input variable. And so ultimately what I'll do is I'll take this input and I'll come back up here so you can see what's actually happening here. And so I'm just going to be assigning based on columns in the input. I'm going to be assigning these values to a variable. So in this case, I'm just taking the value of the first column. In this case, it'd be chromosome 1, or these are just two contigs that are present in this one. And I'm assigning that value to this variable chromosome or chrome. And then here for the start, I'm going to be identifying the minimum across both replicates. So what I'm saying here is the start in replicate 1 less than the start in replicate 2? So this is column 2, this is column 7. And this is kind of an if-and statement. So I'm saying if column 2 is less than column 7, then keep column 2 for the start variable. If it's not, then keep column 7. And a very similar trend here for the end variable, I'm just saying is the third variable or the third column greater than the end of the replicate 2 peak. And if it is greater, keep the third column. If it's not, keep the eighth column, aka the second rep. So then what I do here is I just print the two different variables that I've now created above. And I then, from this print command, it will output whatever it's printing into this new output variable, which I created up here. And so that's pretty much what's going on here. If you wanted to change it to be more restrictive and keep everything that is found across both replicates exclusively, you would just change this to greater than, and you would change this to less than. And that would kind of create the peaks that have been identified specifically in both peaks. You're not kind of creating this union, more of an intersection value. So ultimately, the way you'd run this, again, like everything else, is nohub / make reproducible peaks, ampersand, hit enter. I'm not going to run it now because it takes a little bit of time, but ultimately it'll output everything into this master list directory. And there's some extra other things here I did, but I'll show you what kind of the output looks like here. So in this case, the output will literally just be a text file with three different columns in it, with the chromosome, the start, and the end. And so this is really going to be your consensus set of peaks that have been identified, filtered for signal, and have been identified in both replicates. Okay, now last thing, I swear. So this is kind of specific to this analysis since we're looking at a complex member with multiple different subunits that are actually in this conceptually going to be binding at the same site. So the real question we're asking here is where is the SWE/SNF complex binding? And the way that we're actually going to answer this is by taking the union of the peaks that are observed across three different subunits. So if there's a peak observed in either SMARCA1, SMARCB1, or SMARCA4, we're going to call that a consensus SWE/SNF binding site. And so if you're doing cut-and-run analysis with a particular complex member like EZH2, SWE/SNF, RNA polymerase, or something like that, this is a pretty good idea, taking the union of multiple subunits to identify where this complex is actually binding. And we're being relatively permissive here by taking the union because a peak for one subunit might be called and not for the other two, but it's pretty unlikely that only one of these subunits is going to be binding alone since they're typically found within a complex. So the way that we're going to do this is just a simple bedtools merge command, and we're basically just going to call any peaks that are within 100 base pairs of each other as one singular consensus peak. So I'll kind of walk you through how I'm actually doing this. Really relatively straightforward. So we'll just concatenate all the subunits for a given genotype together. So in this case, we'll take the wild type genotype. So we'll say reproducible peaks, and then we'll concatenate that with the SMARCB1 reproducible peaks and the SMARCE1 reproducible peaks. And we'll just concatenate these all to one particular bed file. SWE/SNF peaks.bed. And so now this is basically you're just stacking each file on top of each other. To help with bed tools, we have to first sort them. So first we'll sort by the first column and make that a priority. So sort by chromosome first. And then we will sort in a second priority the actual start region. And this will sort a second priority and sort by numerical. And so then what we'll do is input this concatenated file, and then we'll output that into a sorted new file. Please sniff peeps sorted.bed. Oh, I added an extra comma. Get rid of that. All right, so now that's sorted, we'll just do a quick bedtools merge, and we will give it the input, which is our sorted wild type also we sniff peaks, and then we'll feed in this option where we're saying if two peaks are identified within 100 base pairs of each other, then we're going to just call that one singular peak. And so this is a little bit permissive, but again, it's unlikely that all three subunits are going to be called at every single binding site. So we're just going to be a little bit permissive here so we get a pretty good idea of where this complex is actually binding. Regions, bed. All right, and so now that if we open this up, we now have a list of regions that our complex is binding to. Um, this is going to be really helpful when plotting signal maps or kind of wild type versus mutant scatter plots. And in the next video, I will in greater detail show you how to actually utilize this bed file to actually create publication ready plots. But that is the end of our analysis for today. I've taken you from the beginning of a bedgraph normalized file all the way to identifying consensus sites in which a complex is actually binding. So if you've made it this far, please, again, like, subscribe, share with all your friends, scientists, anyone who's just learning how to be a bioinformatician. And stay tuned for the next video where we'll be focusing on plotting cut and run data for publications. Thank you, everyone, and have a good rest of your day.